Good afternoon, students. Today we have our first lecture in digestion, and today we will be discussing such subtopics: basic digestive processes and their physiological characteristics, digestion in the oral cavity, reflexes of chewing and swallowing, the role of saliva in digestion, anatomic and histological features of the stomach and its functions, composition of gastric juice and regulation of gastric secretion, motor function of the stomach and its regulation, anatomic histological features of small intestine, and secretory and motor function of the small intestine and their regulation. So let's start. The digestive system consists of the digestive tract, which is a tube extending from the mouth to the anus, plus associated organs, which secrete fluids into the digestive tract. The term gastrointestinal tract technically refers only to the stomach and intestines, but is often used as a synonym for digestive tract. The inside of the digestive tract is continuous with the outside environment, where it opens at the mouth and anus. Nutrients cross the wall of the digestive tract to enter the circulation. The digestive tract consists of the oral cavity, pharynx, esophagus, stomach, small intestine, large intestine, and anus. Accessory glands are associated with the digestive tract. The salivary glands empty into the oral cavity, and the liver and pancreas are connected to the small intestine. Various parts of the digestive tract are specialized for different functions, but nearly all parts consist of 4 tunics, which are the mucosa, submucosa, muscularis, and serosa or adventitia. This will be described in order from the inside of the tube. So the first, the innermost tunic, is mucous membrane called the mucosa. Here you have on the picture, mucosa. The mucosa consists of mucous epithelium, a loose connective tissue called the lamina propria, and the thin smooth muscle layer, the muscularis mucosa. The epithelium in the mouth, esophagus, and anus resist abrasion, and epithelium in the stomach and intestines absorbs and secretes. The second one is submucosa. So submucosa lies just outside the mucosa. It is a thick layer of loose connective tissue containing nerves, blood vessels, and small glands. The submucosa has glands with ducts that extend through the mucosa to empty into the digestive tract. An extensive network of nerve cell processes forms a submucosal plexus or submucosal network. Here you have on the picture submucosal plexus, which regulates secretion from the glands. So the third tunic, the next tunic is muscularis, which in most parts of the digestive tube consists of an inner layer of circular smooth muscle and an outer layer of longitudinal smooth muscle. Another nerve plexus, the myanthetic plexus, here it is, myanthetic plexus, lies between the two muscle layers. The myanthetic plexus is much more extensive than the submucosal plexus and controls the motility or movement of the intestinal tract. Together, the nerve plexuses of the submucosa and muscularis compose the enteric, relating to the intestine, enteric plexus. And the last one tunic, the outermost tunic, is serosa or adventitia. Some regions of the digestive tract are covered by peritoneum and other regions are not. The peritoneum, which is a smooth epithelial layer, and its underlying connective tissue are referred to histologically as the serosa. In regions of the digestive tract not covered by the peritoneum, the digestive tract is covered by a connective tissue layer called the adventitia, which is continuous with the surrounding connective tissue. These areas include the esophagus and the retroperitoneal organs. So the digestive system performs 4 basic digestive processes. There are these 4 basic digestive processes are motility, secretion, digestion, and absorption. So we will start from motility. The term motility refers to the muscular contractions that mix and move forward the content of the digestive tract. Although the smooth muscle in the walls of the digestive tract is phasic smooth muscle that displays action potential induced burst of contraction. It also maintains a constant low level of contraction known as tone. Tone is important in maintaining a steady pressure on the contents of the digestive tract, as well as in preventing its walls from remaining permanently stretched following distension. 2 basic types of phasic digestive motility are superimposed on this ongoing tonic activity. So these are propulsive movements and mixing movements. Propulsive movements propel or push the contents forward through the digestive tract, with the rate of propulsion varying depending on the functions accomplished by the different regions. That is, the contents are moved forward in a given segment at an appropriate velocity. Mixing movements serve a twofold function. First, by mixing food with the digestive juices, these movements promote digestion of the food. Second, they facilitate absorption by exposing all parts of the intestinal contents to the absorbing surfaces of the digestive tract. Next one is secretion. A number of digestive juices are secreted into the digestive tract lumen by exocrine glands along the route, each with its own specific secretory product. Each digestive secretion consists of water, electrolytes, and specific organic constituents important in the digestive processes, such as enzymes, bile salts, or mucus. The secretory cells extract from the plasma large volumes of water and the raw materials necessary to produce the particular secretion. Secretion of all digestive juices requires energy, both for active transport of some of the raw materials into the cell and for synthesis of secretory products by the endoplasmic reticulum. On appropriate neural or hormonal stimulation, these secretions are released into the digestive tract lumen. Normally, the digestive secretions are reabsorbed in one form or another back into the blood after their participation in digestion. Failure to do so, for example, because of the vomiting or diarrhea, results in loss of this fluid that has been borrowed from the plasma. Furthermore, endocrine cells located in the digestive tract wall secrete gastrointestinal hormones into the blood that help control digestive motility and exocrine gland secretion. Next one is digestion. Humans consume 3 different biochemical categories of energy-rich foodstuff, which is carbohydrates, proteins, and fats. These large molecules cannot cross plasma membranes intact to be absorbed from the lumen of the digestive tract into the blood or lymph. The term digestion refers to the biochemical breakdown of the structurally complex foodstuff of the diet into smaller absorbable units by the enzymes produced within the digestive system as follows. So the simplest carbohydrates are the simple sugars or monosaccharides such as glucose, fructose, and galactose, very few of which are normally found in the diet. Most ingested carbohydrate is in the form of polysaccharides, which consist of chains of interconnected glucose molecules. The most common polysaccharide consumed is starch derived from plant source. In addition, meat contains glycogen, the polysaccharide storage from a form of glucose in muscle. Through the process of digestion, starch, glycogen, and disaccharides are converted into the constituent monosaccharides, principally glucose with small amounts of fructose and galactose. These monosaccharides are the absorbable units of carbohydrates. Second, dietary proteins consist of various combinations of amino acids held together by peptide bonds. Through the process of digestion, proteins are degraded primarily into the constituent amino acids, as well as few small polypeptides. And the third one, that most dietary fats or lipids are in the form of triglycerides, which are neutral fats, each consisting of a glycerol with 3 fatty acid molecules attached. During digestion, 2 of the fatty acid molecules are split off, leaving a monoglycerides, a glycerol molecule with 1 fatty acid molecule attached. Thus, the end products of fat digestion are monoglycerides and 3 fatty acids, which are the absorbable units of fat. And the next function of the digestion is absorption. In the small intestine, digestion is completed and most absorption occurs in the small intestine. Through the process of absorption, the small absorbable units that result from digestion, along with the water, vitamins, and electrolytes, are transferred from the digestive tract lumen into the blood or lymph. So let's discuss about the digestion in the oral cavity. So digestion in the mouth involves the hydrolysis of polysaccharides into disaccharides by amylase. No absorption of foodstuff occurs from the mouth. This is important, that no absorption occurs in the oral cavity. Only digestion, only hydrolysis of polysaccharides into disaccharides by the help of amylase. So swallowing is initiated when a bolus or ball of chewed or liquid food is voluntarily forced by the tongue to the rear of the mouth into the pharynx. The pressure of the bolus stimulates pharyngeal pressure receptors, which send afferent impulses to the swallowing center located in the medulla of the brainstem. The swallowing center then reflexly activates in the appropriate sequence the muscles involved in swallowing. The swallowing is the most complex reflex in the body, and multiple highly coordinated responses are triggered in a specific all or none pattern over a period of time to accomplish the act of swallowing. So swallowing is divided into the oropharyngeal stage and esophageal stage. The Oropharyngeal stage or oral stage lasts about 1 second and consists of moving the bolus from the mouth through the pharynx and into the esophagus. When the bolus enters the pharynx, it must be directed into the esophagus and prevented from entering the other openings that communicate with the larynx. In other words, food must be kept from re-entering the mouth, from entering the nasal passages, and from entering the trachea. All of this is managed by the following coordinated activities. So the position of the tongue against the hard palate keeps food from re-entering the mouth during swallowing. Second, the uvula is elevated and lodges against the back of the throat, sealing off the nasal passage from the pharynx so that the food does not enter the nose. Next, food is prevented from entering the trachea primarily by elevation of the larynx and tight closure of the vocal folds across the laryngeal opening or glottis. The first part of the trachea is the larynx or voice box, across which the vocal folds are stretched. During swallowing, the vocal folds serve a purpose unrelated to speech. Contraction of laryngeal muscles allows the vocal folds in tight position to each other, thus sealing the glottis entrance. Also, the bolus tilts a small flap of cartilaginous tissue, the epiglottis, backward down over the closed glottis as further protection from food entering the respiratory areas. Next one, the person does not attempt futile respiratory efforts when the respiratory passages are temporarily sealed off during swallowing, because the swallowing center briefly inhibits the nearby respiratory center. And with the larynx, the trachea sealed off, pharyngeal muscles contract to force the bolus into the esophagus. Now, the esophageal stage of swallowing begins after that. The swallowing center triggers a primary peristaltic wave that sweeps from the beginning to the end of the esophagus, forcing the bolus ahead of it through the esophagus of the stomach, to the stomach. The term peristalsis refers to ring-like contractions of the circular smooth muscle that move progressively forward, pushing the bolus into a relaxed area ahead of the contraction. The peristaltic wave takes about 5 to 9 seconds to reach the lower end of the esophagus. Progression of the wave is controlled by the swallowing center, which was innervation by means of the vagus. If a large or sticky swallowed bolus, such as a bite of peanut butter sandwich, for example, fails to be carried along to the stomach by the primary peristaltic wave, the lodged bolus distends the esophagus, stimulating pressure receptors within its walls. As a result, a second, more forceful peristaltic wave is initiated, mediated by the intrinsic nerve plexuses at the level of the distension. These secondary peristaltic waves do not involve the swallowing center, nor is the person aware of their occurrence. Distension of the esophagus also reflexively increases salivary secretion. The trapped bolus is eventually dislodged and moved forward through the combination of lubrication by the extra swallowed saliva and the forceful secondary peristaltic waves. Saliva, the secretion associated with the mouth, is produced largely by 3 major pairs of salivary glands that lie outside the oral cavity and discharge saliva through short ducts into the mouth. Saliva is about 99% of water and 0.5 to 1% electrolytes and proteins. The salivary sodium chloride concentration is only 1/7 of what in the plasma of that in the plasma, which is important in perceiving salty tastes. Similarly, discrimination of sweet tastes is enhanced by the absence of glucose in the saliva. The most important salivary proteins are amylase, mucus, and lysosome. They contribute to the function of saliva as follows. So first, saliva begins digestion of carbohydrate in the mouth through action of salivary amylase and enzymes that breaks polysaccharides down into maltose, a disaccharide consisting of 2 glucose molecules. Second, saliva facilitates swallowing by moistening food particles, thereby holding them together and by providing lubrication through the presence of mucus, which is thick and slippery. Next, saliva exerts some antibacterial actions by a fourfold effect. First, by lysosome, an enzyme that lyses or destroys certain bacteria by breaking down the cell walls. Second, by a binding glycoprotein that bind to immunoglobulin A antibodies. And third, by lactoferrin, which tightly binds to iron needed for bacterial multiplication. And last one, by rinsing away material that may serve as a food source for bacteria. Saliva serves as a solvent for molecules that stimulate the taste buds. Only molecules in solution can react with taste buds receptors. Saliva plays also an important role in oral hygiene by helping keep the mouth and teeth clean, and the constant flow of saliva helps flush away food residues, foreign particles, and old epithelial cells that have shed from the oral mucosa. Despite these many functions, saliva is not essential for digesting and absorbing foods because enzymes produced by the pancreas and small intestine can complete food digestion even in the absence of salivary and gastric secretion. So now let's talk about the control of salivary secretion. So there are simple and conditioned salivary reflexes. The simple salivary reflex occurs when chemoreceptors and pressure receptors within the oral cavity respond to the presence of the food. On activation, these receptors initiate impulses in afferent nerve fibers that carry the information to the salivary center, which is located in the medulla of the brainstem, as are all the brain centers that control digestive activities. The salivary center, in turn, sends impulses via the extrinsic autonomic nerves to the salivary glands to promote increased salivation. Dental procedures promote salivary secretion in the absence of food because these manipulations activate pressure receptors in the mouth. And with the conditioned or acquired salivary reflex, salivation occurs without oral stimulation. Just thinking about the food or seeing or smelling or hearing the preparation of pleasant food initiates salivation through this reflex. All of us have experienced such mouth-watering in anticipation of something delicious to eat. This reflex is a learned response based on previous experience. Inputs that arise outside the mouth and are mentally associated with the pleasure of eating act through the cerebral cortex to stimulate the medullary salivary center. Stomach, structure and features of the stomach. So this stomach is a G-shaped sac-like chamber lying between the esophagus and small intestine. It is arbitrarily divided into 3 sections based on anatomic, histological, and functional distinctions. The fundus, here you have the fundus. Fundus in the upper part of the stomach lies above the esophageal opening. The middle or main part of the stomach is the body. The stomach muscle layers in the fundus and the body are relatively thin, but the lower part of the stomach, the antrum, has much heavier musculature. This difference in muscle thickness plays an important role in gastric motility in these 2 regions, as you will see shortly. There are also glandular differences in the mucosa of these regions, as described later. The terminal portion of the stomach is the pyloric sphincter, which acts as a barrier between the stomach and the upper part of the small intestine, the duodenum. So main functions of the stomach. The stomach's most important function is to store ingested food until it can be emptied into the small intestine at a rate appropriate for optimal digestion and absorption. It takes hours to digest and absorb a meal that was consumed in only a matter of minutes. Because the small intestine is primarily site for this digestion and absorption, it is important that this speech store the food and matter it into the duodenum at a rate that does not exceed the small intestine's capacities. Second, the stomach secretes hydrochloric acid and enzymes that begin protein digestion. Third, through the stomach's mixing movements, the ingested food is pulverized and mixed with the gastric secretions to produce a thick liquid mixture known as chyme. The stomach contents must be converted to chyme before they can be emptied into the duodenum. So these are 3 main functions of the stomach, but of course it has some other functions which you can read on these slides. Now let's discuss about digestion in the stomach. Although some digestion of chyme and small amount of absorption occur in the stomach is primarily a storage and mixing chamber for ingested food. Secretions from the gastric glands include mucus, hydrochloric acid, pepsinogen, intrinsic factor, and gastrin. A thick layer of mucus lubricates and protects the epithelial cells of the stomach wall from the damaging effect of the acidic chyme and pepsin. Irritation of the stomach mucosa stimulates the secretion of a greater volume of mucus. Hydrochloric acid produces a pH of about 2.0 in the stomach. Pepsinogen is the precursor of the enzyme pepsin. Hydrochloric acid converts pepsinogen to the active enzyme pepsin, which digests proteins by breaking them down into smaller peptides. Pepsin exhibits optimum enzymatic activity at a pH of about 2.0. The low pH also kills microorganisms. Intrinsic factor binds with vitamin B12 and makes it more readily absorbed in the small intestine. So vitamin B12 is important in DNA synthesis and is important to red blood cell production. Gastrin and histamine help regulate stomach secretions. So regulation of stomach secretion is divided into three phases: cephalic, gastric, and intestinal. Cephalic phase, it's the first phase which prepares the stomach to receive food. The sensations of the taste and smell of food, the stimulation of tactile receptors during processes of chewing and swallowing, and pleasant thoughts of food stimulate these centers within the medulla oblongata that influence gastric secretions. Action potentials are sent from the medulla along parasympathetic neurons within the vagus nerves to the stomach. Within the stomach wall, the preganglionic neurons stimulate the postganglionic neurons in the enteric plexus. The postganglionic neurons stimulate secretory activity in the cells of the stomach mucosa, causing the release of mucus, hydrochloric acid, pepsinogen, intrinsic factor, histamine, and gastrin. The gastrin enters the circulation and is carried back to the stomach, where it stimulates additional secretory activity. Second, gastric phase. The greatest volume of gastric secretions is produced during the gastric phase of gastric regulation, The presence of food in the stomach initiates the gastric phase. The primary stimuli are distension of the stomach and the presence of amino acids and peptides in the stomach. Distension of the food wall results in the stimulation of mechanoreceptors. Action potentials generated by these receptors initiate reflexes that involve both the CNS and ENS. These reflexes result in acetylcholine release and the cascade of events in cephalic phase. Peptides produced by the action of pepsin on proteins stimulate the secretion of gastrin, which in turn stimulates additional hydrochloric acid secretion. Moderate amounts of alcohol or caffeine in the stomach also stimulate gastrin secretion. When the pH of the stomach contents falls below 2.0, increased gastric secretion produced by distension of the stomach is blocked. This negative feedback mechanism limits the secretion of gastric juice. The third phase, which is called intestinal phase, so the entrance of chyme into the duodenum of the small intestine stimulates neural and hormonal mechanisms that inhibit stomach secretions. This reduces the acidity of chyme, making it easier for pancreatic and liver secretion to neutralize the chyme, which is required for the digestion of food by pancreatic enzymes and for the prevention of peptic ulcer formation. When the pH of the chyme entering the duodenum drops to 2.0 or below, the inhibitory influence of the intestinal phase is greatest. Acidic chyme in the duodenum inhibits CNS stimulation and initiates local reflexes that inhibit gastric secretion. Acidic chyme also stimulates the duodenum to release the hormone secretin, which enters the blood and is carried to the stomach, where it inhibits gastric secretions. Fatty acids fatty acids and certain other lipids in the duodenum initiate the release of the hormone cholecystokinin, which also inhibits gastric secretion. The small intestine consists of 3 parts: the duodenum, the jejunum, and the ileum. The entire small intestine is about 6 meters long. 2 major accessory glands, the liver and the pancreas, are associated with the duodenum. And the small intestine is where the greatest amount of digestion and absorption occurs. So the secretory function of the small intestine. What is special about this? That the surface of the duodenum has several modifications that increase its surface area about 600-fold to allow for more efficient digestion and absorption of food. The mucosa and submucosa form a series of folds called the circular folds, which run perpendicular to the long axis of the digestive tract. Tiny finger-like projections of the mucosa form numerous villi, and each villus is covered by simple columnar epithelium and contains a blood capillary network and lymphatic capillary called lacteal. The blood capillary network and the lacteal are very important in transporting absorbed nutrients. Most of these cells that make up the surface of the villi have numerous cytoplasmic extensions called microvilli, which further increase the surface area. So the mucosa of the duodenum is simple columnar epithelium with 4 major cell types. First is absorptive cells, which have microvilli and produce digestive enzymes and absorb digested food. Second is goblet cells, produce a protective mucus. Third, granular cells or Paneth cells help protect the intestinal epithelium from bacteria. And the last one is endocrine cells, produce regulatory hormones. The jejunum and ileum are similar in structure to duodenum, except that a gradual decrease occurs in the diameter of the small intestine and the thickness of the intestinal wall, the number of circular folds, and the number of villi as one progresses through the small intestine. So the duodenum and jejunum are the major sites of nutrient absorption. And the last subtopic of today's lecture is secretion and digestion in the small intestine. So the mucosa of the small intestine produces secretions that primarily contain mucus, electrolytes, and water. Intestinal secretions lubricate and protect the intestinal wall from the acidic chyme and the action of digestive enzymes. They also keep the chyme in the small intestine in a liquid form to facilitate the digestive process. The intestinal mucosa produced most of the secretions that enter the small intestine, but the secretion of the liver and the pancreas also enter the small intestine and play essential roles in the process of digestion. The duodenal glands and goblet cells secrete large amount of mucus, and this mucus protects the wall of the intestine from the irritating effects of acidic chyme and from the digestive enzymes that enter the duodenum from the pancreas. The microvilli of absorptive cells have enzymes bound to their free surfaces that play a significant role in the final steps of digestion. Desaccharadases break down disaccharides such as maltose and isomaltose into monosaccharides. Peptidases break down peptide bonds in proteins to form amino acids. Although these enzymes are not secreted into the intestine, the large surface area of the microvilli brings these enzymes into contact with these intestinal contents. Small molecules, which are breakdown products of digestion, are absorbed through the microvilli and enter the circulatory or lymphatic system. So, digestion in the small intestine. Digestion within the small intestine lumen is accomplished by the pancreatic enzymes, with fat digestion being enhanced by bile secretion. As a result of pancreatic enzymatic activity, fats are completely reduced to the absorbable units of monoglycerides and free fatty acids. Proteins are breaking down into small peptide fragments and some amino acids, and also carbohydrates are reduced to disaccharides, and some monosaccharides. Thus, fat digestion is completed within the small intestine lumen, but carbohydrate and protein digestion have not been brought to completion. Special hair-like projections on the luminal surface of the small intestine epithelial cells, the microvilli, form the brush border. The brush border plasma membrane contains three categories of integral proteins that function as enzymes. So first, enterokinase, which activates the pancreatic proteolytic enzymes trypsinogen. Second, the desaccharadases like maltase, sucrose, and lactose, which complete carbohydrate digestion by hydrolyzing the remaining disaccharides such as maltose, sucrose, and lactose, respectively, into their constituent monosaccharides. And the aminopeptidases, which hydrolyze most of the small peptide fragments into the amino acid components, thereby completing protein digestion. Those carbohydrate and protein digestion are completed within the confines of the brush border. So this is all for today's topic, which is the first part of digestion system. And next week, we will be discussing the second part of digestive system. So thank you for your attention and have a nice day. Bye.